Welcome to Decoding the Cell, the Bioanalytical Pipeline. This video explores modern methods of cellular isolation and analysis, moving from surface microscopy to digital proteomics. High-resolution imaging, such as scanning electron microscopy, transmission electron microscopy, and cryo-EM, pushes the limits of observation, offering detailed views of cellular structures and surfaces at the nanometer scale. However, observing organelles doesn't reveal their roles. To dissect function, we must shift from visual inspection to physical extraction. This requires shattering the cell to isolate and analyze its components in their native state. Phase 1 is disruption and homogenate. Cells are broken open using methods like osmotic lysis or mechanical shearing, including high-speed blending or sonication. The process must be maintained at 0 degrees Celsius to preserve enzyme activity and stabilize internal structures. Phase 2 is differential centrifugation. This technique separates organelles based on size and sedimentation rate by spinning the homogenate at increasing speeds. Nuclei, then mitochondria, and finally ribosomal subunits are sequentially pelleted out, leaving pure cytosol. The problem with differential centrifugation is that the batches are often impure. For example, a 15,000 G pellet is a mixed grab bag of organelles. The solution is equilibrium density gradient centrifugation, which sorts components by their intrinsic density, not just size. Phase 3 is finding neutral buoyancy. The mixed fraction is spun in a dense sucrose gradient. Particles migrate until their density matches the surrounding liquid. Pre-treating cells with detergent is a trick used to separate lysosomes from mitochondria and peroxisomes for a cleaner separation. For quality control, biochemical markers are faster and more specific than morphological checks. For example, cytochrome C marks mitochondria, catalase marks peroxisomes, and acid phosphatase marks lysosomes. A hurdle arises when small, membrane-limited vesicles have identical size and density, rendering centrifugation useless. The molecular solution is immunopurification, which exploits unique surface proteins using monoclonal antibodies. Phase 4 is immunopurification. The target vesicle is linked to a monoclonal antibody, which is then anchored to a heavy material like S. aureus bacteria. This heavy complex is then easily captured via low-speed centrifugation. The final hurdle is obtaining a complete inventory. Historically, technology could isolate organelles but not catalog every component. The digital solution, digital proteomics, uses mass spectrometry to map every protein simultaneously. Phase 5 is proteomic sequencing. After high-purity isolation, the organelle's polypeptides are cleaved by trypsin. Mass spectrometry then determines peptide mass, mapping it against the genome to identify origin proteins. This can reveal hundreds of distinct proteins. The modern cell analysis pipeline integrates observation, disruption, fractionation, targeting, and digital mapping. Overcoming the limits of observation requires this integrated pipeline of physical disruption, molecular targeting, and digital synthesis.